specific. I'm going to start with a short movie, but the guys remotely off-site can't hear the sound, so I'm going to add a little narration. You're going to hear both. But anyways, it's about this remarkable new gene therapy treatment for a disease called Metachromatic Leukodystrophy. It just shows where we're going with these remarkable therapies for these inborn errors. So basically.
Children with late infantile onset metachromatic leukodystrophy lose the ability to walk, talk, swallow, eat, and see. Most kids with MLD won't survive beyond their 5th birthdays. In 2010, using stem cells derived from their patient's blood, researchers pioneered a treatment to repair the defective gene that causes MLD. Giovanni Price was the second child in the world to undergo this treatment. Today, Giovanni is 5 years old and has no symptoms of MLD. On August 24, 2015, Giovanni Price did something no child with this disease has ever done before. He walked to school for his first day of kindergarten.
So he's broken all the milestones
when he was born,
but we don't know how long this treatment's going to last, right? But it looks good so far.
By the age that he is right now, should be close to the end of his life. And so we see the complete opposite. I mean, we see him going to school and making friends and riding his bike in the driveway. And the doctors don't tell me he's, they've never used the word cure. They've never.
So he's obviously not cured, right?
They don't know. I mean, we don't know, but I hang on to those words that he's going to continue to grow up just like any other 5-year-old. My hope is that gene therapy combined with
gene therapy and newborn screening, catch it early.
Will
that's the ticket.
Save other families from going through what we went through and what all of our friends, our MLD friends, go through with their kids. He has no idea that he's a miracle. He has no idea.
Yeah, so I'll come back and talk about this therapy. It's not developed here, it's from Italy, but it's remarkable. I thought you should hear about it so you know what's coming in advanced kind of therapies for these diseases. My disclosure statement, here it is. I was kind of a bad kid when I was in high school, I got in trouble and all of that. Oh, sorry, you want to hear about my relationship with, yeah, I lied to the police when I was 10 years old. It was a big mess, yeah. So my sister was there, she lied too, so I'll never forget that. But I grew up to be a pretty good guy after that, so anyways, that's my disclosures. So newborn screening for treatable genetic diseases. And I have an endowed chair position. That doesn't mean I'm chair of the Department of Chemistry. You would have to you would have to torture me for 10 years to get me to agree to become a departmental chair. So I have an endowed chair. That doesn't mean I'm chair of chemistry, right? Just to make that clear. I don't have any skills to be a chairman of a major chemistry department. So what is newborn screening? I think most of you know, but I'm going to talk about just the gist of that, and then talk about these lysosomal storage diseases, which I think many of you know about, but just so we're on the same page. And I'm going to talk about this new technology that we've developed here with Ron Scott and others on screening for these diseases. I'm going to show you some pilot studies. I'm going to talk about this thing of diagnosis and prognosis. You'll see what I mean by prognosis in a minute. And I'm going to talk about a very exciting chapter coming as a collaboration between me and Si-Hoon Han at Seattle Children's Research Institute on newborn screening versus proteomics, which is kind of the marriage of mass spec and newborn screening. People like Andy Huffnagel are trying to apply proteomic techniques in clinical medicine, in cases where immunoassays don't work very well. And I'll talk about that in the context of newborn screening. So just to give you a quick story on newborn screening, I think you know about galactosemia, but this is lactose, and it's not clear why we're born and we eat all this lactose. It's in mother's milk. It's not the perfect food, right? Because it has one galactose and one glucose, and yet we need 100 times more glucose than we need galactose, right? Because glucose is our energy. It's like the gasoline of the car. Galactose is like the fender of a car, the structural component. It's not clear why, we need a lot more gasoline than we need fenders. So the liver comes to the rescue, and I don't know why we're designed to eat food that needs to be rescued by the liver, but I think it has something to do with the bacteria that grow in our gut, and the good guys live on lactose, and we want the good guys down there, and maybe that's it, but nobody knows. The liver comes to the rescue, and it has a 3-step enzymatic pathway to convert all of this excess galactose to glucose. And if you have a mutation in that process and it's inefficient, you die of galactose poisoning, whatever that means. I suppose it means you put galactose in places that you should put glucose. And it's very severe disease, and it's fairly simple to treat just by reducing dietary intake of galactose, right? So we've been screening for this all over the world for many years, and if we get a hit, it's an emergency. We call the family, get the baby off mother's milk. It's, total irony. You're sort of killing your baby with mother's milk, cow's milk, doesn't matter. We got to put you on a special formula. But if we didn't do that, and this is a rare disease, right? So there's very little family history. These are autosomal recessive diseases. Your husband and wife come from different parts of the world. Typically, they have no family history. These two come together and have a kid with 2 bad genes, and you have a autosomal recessive inborn error of metabolism. So we have to just screen everybody in the population in a non-biased way to find the rare case of a baby, and then we can treat it early. And if we wait until the baby gets too sick to end up, at Northgate Mall, and then Seattle Children's, and then another Children's, and by the time they figure it out, the baby is too far gone and you can't treat. So if only we had known about this, we could have fixed it early. It's a no-brainer, right? So about 1 in 40,000 babies are born with mutations in a DNA that leads to a defective enzyme for converting galactose to glucose. When this happens, we get an inborn genetic disease called Galactosemia. It's rare. It can take several months to diagnose. I think you know all this. And by the time the diagnosis is made, it's too late to save the baby. If only we had known at birth, we could have fixed it, or partially fixed it. The only reasonable solution is to screen everybody in the population. Okay, so all sophisticated countries around the world do this. Australia, Europe, United States, Japan, China. This guy, Robert Guthrie, it's an interesting story. His niece is head of Virginia Mason Research Institute here in Seattle, Jane Voigt, and I work with her husband, Fred Buckner, on parasites. So anyways, this guy, Guthrie, invented the concept of newborn screening through these dried blood spots on newborn screening cards in the 1950s. Here's a family picture in Seattle. I've met the family over the years. He's no longer with us, but he started the concept of newborn screening for PKU, which I think you all know about, phenylketonuria, and this idea of dried blood spots on newborn screening cards. He was at Buffalo, University of Buffalo. So typical numbers, 1 in 100 mothers might carry a typical mutation for a genetic disease. Let's say 5 of you in the audience. Well, the last time I gave this talk, there were 500 people in the audience. That was the university faculty lecture. So let's say 2 of you in this room. What am I talking about? 1 of you in this room is probably a carrier for galactosemia, and then the father comes along, the same deal. So 1 in 100 squared, is 1 in 10,000, and if 25% of the offspring will have 2 bad genes. So that's how you get 1 in 40,000. I think you all know this. So cystic fibrosis, right? 3,000 babies. 1 in 3,000 babies born with cystic fibrosis, the most common genetic disease, I think. Serious genetic disease. I mean, it's like 1 in 8 or 10 people are carriers. It's amazing when you think about it. So millions of babies are born each year. We screen for about 40 to 50 individual disorders. According to the CDC and the American College of Medical Genetics, it's one of the most successful public health programs of all times. And 1 in 300 babies, this many per year in the United States, out of 4 million babies, are saved as a result of newborn screening. So this is a good thing. I mean, and a lot of the low-hanging fruit in this area has been done, like PKU. We should screen for that. We have a good treatment. It's a serious disease. Galactosemia is a no-brainer. But now we're getting into areas that are more controversial, right, as you'll see. Diseases that the treatments are very expensive. The treatments don't cure the baby. They make the baby better, but far from perfect. There could be late onset variations of these diseases that scare families. It's not so simple, as you'll see. Every state more or less has a newborn screening lab. It's like the state police are involved, and when the FBI show up, the state police say, go home, it's our stuff, right? So the FBI tries to influence the state police, but the state police doesn't really want the FBI there. That's exactly how to describe newborn screening. It's a state-by-state thing. The feds try to stay out of it, but it's, as you'll see, it's a little more complicated. Every state has one. Every state has a director. Here's John Thompson in Shoreline, runs the newborn screening lab for Washington State, okay? So we hear a lot about next-generation sequencing. I just want to have one slide about this. This is a no-go for first-tier newborn screening. Some genomic people might disagree. I think they're wrong. Here's the deal about, so what, pretty soon, I don't know, pretty soon enough, we're going to be sequencing the genome of lots of people, lots of babies. When are we ready to use that information for high confidence newborn screening or prediction of disease onset? The $100,000 question in this field is nobody knows. Is it 10 years from now? Is it 50 years from now? I don't know. It depends on the disease. It's a very complicated question. It's a very big question, okay? So the DNA sequence, if you think it tells you everything, you're wrong, right? It doesn't. Well, first of all, it's still too slow and expensive for first-tier population-wide newborn screening. But that's not the major problem. Even if we can make it $1 per baby, which would be the target price range, and super fast, that's not the major problem. If you have severe mutations, the so-called low-hanging fruit, like premature stop codons, frame shifts, insertions, deletions, you still have the phasing problem, right? You still don't know if you have both mutations on the same allele or one from each parent, then you got to go sequence the parent. We can do that, but there is a phasing problem. But next, large deletions might be missed if you have one large deletion on one allele, not on the other. You're just going to read a good sequence. You're not going to see the allele unless you do gene counting, which you don't really do. You're going to miss large deletions. We have variations of unknown pathogenic significance, the so-called VUs. This is not so uncommon. So if I sequence Ron Scott's DNA, his DNA is going to differ from mine in one gene in about 1 in every 50 nucleotides. But if I look it up in a table of 60,000 humans, I'm going to see that his variations are well known in the population, as are mine. And so I'm going to ignore his differences between him and me because I see them in so many humans that don't have a disease. But once in a while, not so infrequently, you find these rare mutations that are not found in 60,000 humans, and we don't have any biochemical information about these missense mutations, and so they're officially unannotated and they're variations of unknown significance. And this happens a lot. We can't ignore these because we don't know if they're bad guys or good guys. We know they're rare guys. The biggest problem in my mind is what I call the ABC problem. There's probably another name for this. Here's an example. A patient presents with a genotype A/B, okay, a set of mutations A on one allele and B on the other. And A and B are thought to be partially penetrant mutations. Mutations that cause less than super severe disease, maybe later onset disease, okay? Remember, diseases are spectrums, so are mutations. Then we find a new patient, BC, also late onset. And so we put A, B, and C into the database of known partially penetrant pathogenic mutations. Then comes along not AB and not BC, but an AC. So let me ask the audience, is AC guaranteed to have this late onset disease if AB does and BC does? What do you think? How many people say, yes, this person is expected to have the disease? How many people say we don't know? Yeah, so you know this ABC problem. The answer is we don't know. So it's a numbers game. You're adding combinations of partially penetrant mutations, and the numbers matter. So not all pairs of partially penetrant mutations will lead to disease, and we only see a tiny fraction of the combinations of partially penetrant mutations, right? So if we have 200 partially penetrant mutations, there's 200 squared combinations, and we only see a few hundred. We're missing lots of data on all the combinations. And that is the biggest challenge in next generation sequencing for first tier newborn screening, in my opinion. Not the cost, not the time, not the phasing, not the VUs, but the ABC problem. And time again in newborn screening programs, you see this ABC problem haunting the use of next generation sequencing for first tier newborn screening. Okay. Lysosomal storage diseases. Family of serious genetic diseases, many of which can be treated. One example is MLD, you saw in the video. It's caused by mutations that lead to a dysfunctional lysosomal enzyme. Without screening, diagnostic odyssey is a problem, often too late to start treatment. And expansion of newborn screening programs worldwide to include a subset of these diseases is a very big topic of discussion, and in fact is occurring right now, thanks in part to the technology that we developed here at University of Washington, for better or worse. So just to tell you a little bit about how these diseases are treated, you probably know about enzyme replacement therapy, but I'll go through it quickly. So all the proteins in the cell are recycled. Why? Because I guess they poop out, right? They're not that stable. Some last a few days, some last a few hours, some last a few weeks. They wear out. They have to be recycled. Typically, they're broken down and reassembled. So in the cytosol, you have things like the proteasome that's chewing up all the proteins and recycling them. But what about the proteins on the cell surface? They're not in the cytosol. How do they get recycled? They get recycled by enzymes in the lysosome. And not only proteins, but glycolipids on the cell surface, sphingolipids, proteoglycans. So it turns out it's a magic trick. So how do these proteins on the outside of the cell go through the plasma membrane and go through the lysosomal membrane to come in contact with the enzymes inside the lysosomes? So they have to go through two membranes, and they do that without going through any membranes. It's a magic trick. It's like the ring trick, where you take apart two rings, and so the way it works is it's a magic trick. So not really. The proteins, you get endocytosis. This vesicle seals off into a vesicle where the proteins now on the outside of the cell are now on the inside of the lumen of the vesicle. And this guy, like a soap bubble, fuses with this guy, another soap bubble, so that you get one bubble, and what's inside each bubble becomes inside the same bubble. So it's just fusion of membranes. And now the enzymes that were on the cell surface are in contact with the lumen of the lysosome and the enzymes inside. So I'll tell you, so lysosomal storage diseases, there's about 40 different enzymes required for recycling of all these goodies on the outside of the cell. If we have DNA mutations that lead to dysfunction of any one of them, we have a specific lysosomal storage disease, often with different symptoms. So cells die for different reasons because of the accumulation of these metabolites. So enzyme replacement therapy is pretty simple. We inject the recombinant enzyme that we make in a factory into the blood of a patient. In the interstitial fluid, it can come in contact with many cells of the peripheral tissues, not the CNS, because the enzyme doesn't go across the blood-brain barrier, as you know. But it can just, as a bystander, be taken up by endocytosis in the fluid phase and delivered to the lysosome from the outside. So we can just insert lysosomal enzymes from the syringe needle into the blood, into the interstitial fluid, and into the lysosome of cells that are in contact topologically with the interstitial fluid via the blood. So we just give you the missing enzyme, okay? And if we could get these enzymes across the brain, into the brain, we could do this for brain diseases, but we don't know how to do that very well. People are working on sort of Trojan horse type approaches, but nothing in the clinic yet. All right, enzyme replacement therapy. You saw the movie maybe. You probably didn't see the movie about actually our president mentioned John Crowley, played by Brendan Fraser, at his first speech. I didn't show you the movie because, you know. He honored John Crowley, who, it was Rare Disease Day. You can watch the movie on YouTube. John Crowley had 2 kids with Pompe disease. He was a businessman. He quit his job. He found the scientist, Harrison Ford, right? This is the first and last movie directed by Harrison Ford. I think 3 people saw this movie, and they all worked for me. We went to the Varsity Theater. They said they were showing it. We got there. They said they changed their mind. We had to drive out to like Auburn, Washington. We stopped at the Muckleshoot Casino. And we got to Auburn. There was like 1,000 people in line to see Avatar. And we went to see this. And it was really funny because the business guy was yelling at the scientist, like, how come you're not working fast enough? And I turned around and looked at all my graduate students and I said, yeah, see, that's what I'm talking about, right? And anyways, it's a true story about this guy that started a clinical trial and got his kids treated, and they're doing pretty well, these kids. You can watch the YouTube. It was kind of a remarkable story about the treatment for Pompe disease. All right, so what about if the thing is in the brain? Like MLD, the brain disease. So let me tell you just briefly how this movie thing works. They drain about, this is in PhD terms, not in medical terms, right? They drain 1 pint of baby's blood from the placenta through the umbilical cord. This is called cord blood, right? So think of the umbilical cord as Tygon tubing attached to this big bag of blood that's full of, you can't take 1 pint of blood out of a baby, right? Even a PhD knows that, right? So they take a lot of blood out of a placenta through the umbilical cord, and it's just baby's blood. The fact that it's cord blood is just plumbing. Nothing special. They isolate immature stem cells. They're supposed to be in the bone marrow, but they're floating around in the baby's blood. And they, by magnetic cell sorting, and they're going to engineer these cells to overproduce the missing enzyme. So they take an engineered HIV virus, a lentivirus, just garden variety HIV virus, and they engineer it so it's not infectious. And they insert a copy of the gene for this enzyme with a very strong viral promoter, CMV promoter if you care, into the genome semi-randomly of these stem cells, hopefully without causing cancer. Then they destroy the baby's bone marrow with chemotherapy and radiation, and they do an autologous bone marrow transplant. They give these engineered stem cells back to the baby. They go into the bone marrow, repopulate all the blood cells. So what does this have to do with the brain? So obviously you can treat blood diseases this way. But it turns out the immune cells, some of them have to get into the brain because you have macrophages and dendritic cells and all kinds of things from your bone marrow living in your brain. So they burrow through the blood-brain barrier or the capillaries, and they reside in the brain. These lysosomal enzymes, they can get into cells, but they can also get out of the cell, just reverse the process I just showed you. So the idea is that these cells in the brain that came from the bone marrow that are producing large amounts of the enzyme through the gene therapy to augment the process are spewing into the brain interstitium good enzyme, which are then taken up by cells that did not come from the brain, from the bone marrow, by endocytosis. So this is called cross-correction, where one cell feeds the other cell a lysosomal enzyme through the culture medium. So this cross-correction combined with overproduction of enzyme is the basis for this stem cell autologous bone marrow transplant with gene therapy that you saw in the movie, developed by Alessandro Aiuto's team at the University of Milan, a very famous science paper a few years ago. I think some of the first applications of gene therapy in human health will be lysosomal storage diseases. So we need newborn screening for MLD. I'll tell you how we do that in a minute. All right, so now let's talk a little bit about how we do all this screening, and it's going to come down to mass spectrometry. So you can do fluorescent assays for so here's the deal. We don't want to do DNA. You know, we could measure the abundance of the lysosomal enzyme by some sort of an ELISA. because most mutations that cause disease lead to a non-stable, misfolded protein that's degraded, and therefore the protein abundance is usually low. So why don't we just measure the protein abundance? We could do that, but we would miss cases where the mutation leads to a dysfunctional enzyme and yet a stable protein that's not degraded. So we would have a false negative problem. We don't know how bad that problem is. Maybe we would miss 5 or 10% of the cases. Maybe that's too much. But we decided 15 years ago to go after the function of the enzyme, not the abundance of the protein and not the DNA sequence, the activity of the enzyme. So now we want to measure a bunch of enzyme activities at the same time in a dried blood spot. And mass spectrometry is the way forward for lots of reasons. You can do many enzymes at once, as you'll see. You don't always have a fluorometric assay for your enzyme of interest. Sometimes you do. So it would be nice to be able to do this in general, not just for which you can do fluorescent assays. And mass spec, I'm not going to go into it, is more precise than fluorescence and gives less false positives. Mass spec is already used in newborn screening lab, so all your amino acids are measured by mass spec right now. That's how they detect tyrosinemia. That's how they detect PKU, et cetera, et cetera. So mass spec. And Ron Scott got very excited about this when I mentioned this to him about 15 years ago. So how do we do many enzymes at once? Well, it's easy. We just take a bunch of substrates, we put it in the blood spot, that has the enzyme. We have a bunch of enzymes convert to a bunch of products over time, and after a few hours, we measure the amount of product. Well, how do we measure the amount of product? I mean, we count them in the mass spectrometer. Just 1, 2, 3. We have a detector. So we have a machine that separates molecules based on the mass. So we send all the products with this mass through and hits the detector, and it goes ping, ping, ping. So we count the molecules to determine the amount of product. Divide by the time is the enzyme activity. Okay, but we don't know if the machine is telling the truth. I mean, it gives us ping, 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 but maybe it missed a few pings. So we include an auditor. And it's not a once-a-month auditor. Every experiment gets an auditor. Okay, so there's no cheating. We'll catch you if you cheat. So how do you do molecular auditing? It's very simple. With mass spec, you add exactly the same product chemical, but made heavier with deuterium instead of hydrogen. So if you miss a few of these, you miss a few of these because they're the same chemical, right? Right? It's like if you're counting jelly beans in a jar and you want to know how many red ones. And you put some 5 red jelly beans in the jar that are injected with garlic. And you say you've got 12 red jelly beans in the jar, and then you taste them afterwards, and you taste 4 of them taste like garlic. But you put in 5, so you know you missed 20%. That's exactly what you can do with an internal standard. Think about it that way. It's an auditor. So this becomes perfectly quantitative. In fact, the most quantitative clinical technique I can think of. We use dried blood spots. So we take a 3 millimeter punch of this dried blood spot. We mix it with a buffer with a bunch of different substrates for a bunch of different enzymes. We incubate to allow the substrate to convert to product. We extract into organic solvents just to get rid of the buffer salts, which kind of interferes with the mass spectrometry process. And then we inject this into the mass spec. And the mass spec is just a tube that allows ions to go through with a certain mass. So we look for the needle in the haystack just by selecting on certain masses. Okay. Now I can tell you about how we know the thing we see is really the product of interest, and I can show you all the controls and everything, but there are ways to do that. For example, if you leave out the substrate and you don't see the mass spec signal for the product, you know the product signal came from the substrate. It didn't come from some ghost peak in the blood, right? You leave out the substrate, you put in the blood, you don't see the product, you need the substrate, it must be the product, right? That's the idea. So here's one of the substrates. Never mind all the details. It's some enzyme recognizable sugar, and then we make this product where we can put heavy isotopes to make a chemically identical. See, these are the same thing. This internal standard just has deuterium. Without going into details, the substrate, what we insist, of course, is that the substrate detects the enzyme of interest. If we detect multiple different enzymes with our substrate, it's no good. So sometimes we need to add inhibitors of off-target enzymes. We can do that, but the assay is only as good as there be a one-to-one correspondence between the substrate and the enzyme of interest. I think you all know that. Just to show you the spectrum of substrates, these are sphingolipids. If you can't take the phosphocholine off of sphingomyelin to make the ceramide, you have Niemann-Pick. You can't take galactose off, you have Krabbe. You can't take glucose, you have Gaucher. These are ceramides. How do we know the ceramides came from these substrates? Because you have ceramides in your blood. Because the R groups on these ceramides are a little bit shorter than the natural ceramides, and so we asterisk these ceramides to come only from these substrates via the enzyme of interest. So we're sure that the amount of this somewhat artificial ceramide came from our enzyme of interest, okay? The easy tricks. We have glycohydrolyzing enzymes. We have sulfatases that take sulfate. Anything you want, we can do by mass spec, okay? We have a kit, FDA-approved kit from PerkinElmer to do 6 of these. Rona mentioned some states. About 30% of the United States is now doing this live for newborn screening for Pompe disease and MPS I. Washington State's going to start next year. People at Children's Hospital are getting ready. Taiwan is doing this like crazy. Japan is starting. Australia. Europe, not so fast. They're a little bit anti-screening over there. I don't know. You can talk to them. Now, here's the ultimate solution. So I want to talk to you about this megaplex. It's developed by Xinying Hong, who's sitting somewhere over there, and you'll see her later. We can do like 15 enzymes and 5 biomarkers at the same time inside 2 minutes by mass spec. So I think we could do 200 enzymes, but I always tell Rana, why don't we screen for everything? And she tells me, because we should only screen for things that we can treat. Otherwise, you don't want to know. And I agree with that. 10 years ago, I didn't know that. I wanted to screen for everything. And she sort of set me straight. I'll never forget that conversation. But anyways, do we want to screen for some genetic disease that we can't treat? Well, if the first kid dies, you probably want to know why, so you can be on the lookout for the second kid. But other than that, I don't know, probably not. Although it's coming through genomics someday. Anyways, we have a choice, actually. We can look at the enzyme activity, which I've been talking about, which amounts to the amount of product made as a function of time. Or the other way we can do this, which makes a lot of sense, is if you're missing an enzyme, the substrate for that enzyme should accumulate. It's not always that simple, but for some diseases, the only choice is to look for the accumulated substrate as a result of a deficient enzyme. So that becomes a biomarker, just to measure the level of the substrate. We do it all inside of 2.4 minutes by LC mass spec. We look at 15 enzyme products with 15 internal standards, and we look at about 5 biomarkers also with internal standards. Everybody has their own auditor. There's no cheating. There's no lying. It's the real deal. It's not fake news, right? It's real. We can do this beautiful multiplex, and I won't go into it, and we can add pretty much lots of things that you want to this. We did a pilot study recently in the Washington Newborn Screening Lab. So you can't just turn this thing loose and go, you have to do kind of a clinical trial. So if you expect MPS II disease, for example, you expect 1 in 100,000 patients. And if your screen gives you 10 per week, you have a serious false positive problem. You're going to be pulling your hair out, sequencing all these DNA, scaring all these families, all these false alarms, a lot of sleepless nights. Big mess. The system can't handle it. The families can't handle it. So the false positive rate has to be manageable. The best false positive rates we have are typically, 1 in 10 screen positives turns out to be the real deal. 9 out of 10 turn out to be false alarms. That's like really good, okay? I'm talking about false positives of like 1 true positive in 1,000 false positives would be a mess. So we're always going to have to do second-tier tests to reduce the false positives, but when it gets so bad, we can't handle it. So we have to do a pilot study. So we ran Let me show you some numbers. We ran a pilot study for 5 different mucopolysaccharide doses in the Washington State lab recently. We went to 100,000 de-identified samples, so we can't identify these patients, we can't offer them treatment. We just do a research study. We're not allowed by law to identify the patients. It's kind of too bad, but that's IRBs and paperwork and all that stuff. It's against state law to do a prospective pilot study. We need parent consent, and there's lots of places you can have a baby, so Ron and I aren't going to run around asking all these parents for their consent. Although we thought about it, but it's just not practical. So we screened 100,000. We had 20 screen positives. That just means the enzyme activity is below our cutoff. You can argue, what is our cutoff? When it's low, it's a screen positive. The enzyme is low, we're worried. So we had 20 hits, and we genotyped all these, and 2 have genotypes consistent with MPS II. We're pretty sure these 2 kids will develop MPS II. It's an X-linked disease. 2 mutations are very severe, well known in the database. So 2 out of 20. We had 1 MPS IIIB hit, genotype normal. 8 MPS IVs, 2 probably MPS IVs, 5 MPS VIs, all normal. We had, this one's really remarkable. MPS 7 is thought to be 1 in 300,000, so really rare. And we got one in the first 100,000, and the kid showed up at Children's Hospital with MPS 7. So you can see the numbers. We have about, for 100,000, 5 diseases, we have about 30 hits. About 4 or 5 of them are true cases. And that is remarkably successful pilot study. So based on this, we hope to put some of these diseases forward for widespread more screening. And there'll be more pilot studies of this type coming in the next few years. In New York, we're going to be doing a prospective pilot study where we can consent 3 large hospitals in New York City and get 45,000 babies per year with parent consent. So if only it were so simple. The enzyme's high, you're healthy. The enzyme's low, you're not healthy. It's not that simple. It's a continuum, right? I think you all know this. So you start off with normal enzyme activity, call it 100%, population normal. As the activity gets smaller and smaller, we generate a continuum of events. For many enzymes, we typically have more activity than we really need, and so even if the activity drops to 20 to 30% of normal, we're still healthy for our lifetimes. Then we get into the so-called pseudodeficiency regime, where the activity drops further, let's say to 10%, but you still don't get a disease. That would be a pseudodeficiency, but you can see it's a continuum. What is normal and what is pseudodeficiency? And what is low and what is high? But officially, a pseudodeficiency is a mutation, a mark in the genome that causes a reduction, a significant reduction in enzyme activity, but not to the point that it causes pathogenicity. Okay, but it is a broad definition that has lots of interpretations. So it's kind of messy. So when the activity gets below pseudodeficiency, we have diseases that are late onset, right? We have diseases that start as teenagers or adults, and then when it gets even lower, we might have infantile diseases. And when it's even lower, we might have a miscarriage, right? So it might not be viable. So it's a continuum, right? And so it's not black and white. And we struggle with this. For some diseases like pseudodeficiency, like MLD, the pseudodeficiency is a huge problem. 1 in 7 Europeans carry a common mutation in their genome that reduces the activity more than 10-fold. So 1 in 7 squared. So 2 in 100 people will read very low enzyme activity and yet never develop MLD. The difference between these people and somebody with MLD is like MLD have less than 1% activity. Homozygous for the pseudodeficiency, you might have 5%. So we're going to be splitting hairs at the low end to try to figure this out by enzyme activity. It's not possible. So the only way forward is to look at the accumulated substrate for MLD, which we can do in this multiplex assay. The accumulated substrate is the so-called sulfatide. This disease is caused by a deficiency of an enzyme that takes the sulfate off of the galactose on the sphingolipid. This is a big component of myelin, and these patients lose their myelin. It's a leukodystrophy, right? A white matter loss. So we measure accumulation of sulfatide substrate in dried blood spots by LC mass spectrometry. I was very nervous because sulfatides are maybe 5 to 10-fold elevated in urine, but we don't collect urine for newborn screening. I always wonder what's the big deal, but I'm told that collecting urine for newborn screening is off the table in the United States. They do it in Quebec, not the rest of Canada. Historical thing, I don't know. But what's the big deal collecting urine spots from babies? Just, I hear it's a big deal and it's not gonna happen. So we then turned to blood spots and we found that sulfatides were elevated in newborns with MLD, but not very much. So how do we know sulfatides are elevated in newborns with MLD? So a kid is born, he's healthy. He or she develops MLD when they're 3 years old. And how do we know what their sulfatide levels are at birth? Because we don't know they have MLD until they're 3 years old. Well, if we go to Kentucky, that's a problem because they throw away the blood spots at birth a few days after the kid's born. I don't know what's going on. In California, they store them for 20 years. That's a good thing for research, so we can go back and retrieve those newborn blood spots from the freezer of California for those 5 kids over the last 10 years who developed MLD, and we can get a glimpse of their sulfatide in a newborn blood spot. And that's what you see in red. And this is a typical week of data for 1,500 random newborns, and you can see that the elevation of sulfatides at birth in MLD is 0.3 versus sort of approaching 0.2. So this seems like a close call. Our best estimates are that we're going to get 0 to 2 or 3 false positives per week, which is probably too many, be 100 per year. So we're scratching our heads about what a second-tier assay might look like to stratify these hits. But we think screening for MLD will be possible this way, but it's going to be a little bit challenging. Okay, this shows you what we're up against. Where we can't do the enzyme activity, we have to do, let's say, a minor increase in sulfatide levels in blood spots. All right, now I want to turn to this diagnosis prognosis thing, which is going to, children's hospital people going to be interested. So what happens often in newborn screening is we get a kid with low enzyme activity. How low is low? Well, we're worried about it. We don't know. Maybe the kid's not sick yet. When are they going to get sick? So they're not sick yet. When are they going to, if and when they're going to get sick? That's not diagnosis. I think that's called prognosis. When are they going to get sick? I don't know. When are they going to get sick? Maybe never. Maybe in 5 years. The parents kind of want to know. The doctors kind of want to know. The kid kind of wants to know. They were found by newborn screening, which we didn't do before, so that's kind of a mark against newborn screening, the fact that we're putting these families into this kind of horrific situation, right? It's not, I don't know, right? You found out that your kid might get MLD. Not so good. Now, all the parents with MLD kids, they think, let's screen, because the only chance you can save these kids is newborn screening. But all the kids, families that don't have these kids, they say, well, wait a minute, I don't know. They're not sure. If I ask you, do you want to know? I think you don't know whether you want to know. If I ask you in a week, I still don't think you know whether you want to know, right? I don't think you'll ever know if you want to know the answer to this question. I don't know. You might know. I don't know. Ask me. I don't know. So when do you want to know? If do you want to know? If the answer is maybe. So there's a big problem, right? I think you get the idea. And as we screen for more and more things, it becomes more and more controversial, right? That you're going to get this when you're that old, and maybe, but we don't know. My hypothesis is that the amount of enzyme is important. So the lower the enzyme, the earlier you're going to get it. I mean, that seems to make sense. But people have sort of gone against that hypothesis because I think the assays are not sensitive enough to see the difference between 1%, 2% kind of numbers. But with mass spec, you can do it for the first time. So if you have at risk for Krabbe disease from newborn screening, you go to David Wanger's lab where he measures the Gal-C enzymatic activity in leukocytes. He cleans it up a little bit by using leukocytes instead of a dried blood spot so he can normalize for the amount of protein. But he uses a radiometric assay, Can he tell the difference between, let's say, 1 and 2% versus 2% of residual, 1 or 2% of normal activity? Okay, let's say population mean is 100%. Can he tell a factor of 2 between 1 and 2%? The answer is no, because in his radiometric assay, the difference would be like 5 dPMs. No, he cannot. Statistically significantly, he cannot tell the difference between 1 and 2%. But with mass spec, we can. Now, how do I prove that? It's easy. I take blood cells, immortalized leukocytes from a patient with Krabbe disease that has no Gal-C activity because they're missing the gene. They have large deletions in both copies. And I mix in various amounts, preconceived. I determine the amount of normal leukocytes, and so I determine the x-axis, the percent of normal activity. And you can see at 1% activity, I get this with 2 micrograms of protein or 5 micrograms, and at 2%, I get that. And you can see the error bars on triplicates. So I can tell the difference between 1 and 2% unambiguously. In fact, I can tell the difference even lower. To make a long story short, we've looked at this assay on a handful of screen-positive patients, and we're doing pretty good with prognosis. We can tell you the kids that are at highest risk to develop Krabbe disease, and so far they're developing symptoms in a few years of life. And we can tell the group that is at less risk, and so far they haven't developed Krabbe disease out to 15 years. So we're getting better from measuring the residual activity very accurately in leukocytes to follow up on newborn screening. We're getting better at prognosis. And we're never going to get perfect, right? But we're trying to do better with very precise mass spectrometry assays that you can't do by other methods. Just to end in the last 5 or 6 minutes, I know Rona's very interested in lysosomal acid lipase. Let me tell you a little story that's kind of cool, kind of proud of it. At Children's Hospital, they do a lot of assays for this enzyme. If you're deficient in this enzyme, you have cholesterol ester stores disease, or also called Woman's disease, or it's a different type of lipase deficiency. The standard assay in the field is a fluorescent assay with a 4-MU palmitate. And the problem is there's many esterases in blood that can hydrolyze this fluorometric substrate. So what they do is they do 2 assays, one without and one with an inhibitor that's selective for the lysosomal acid lipase. And the difference between the two, the lysosomal acid lipase inhibited, not inhibited, is the lysosomal acid lipase component to the total serum esterase activity on the substrate. So they do 2 assays in parallel. But this is not good for newborn screening because you're going to be subtracting 2 large numbers with errors, and you're going to end up with a small number with a big residual error. So I don't think it's going to work for newborn screening, and I won't bother you. You know how to propagate errors. But anyways, so the first thing we tried, we went back to the natural substrates of this lipase, which are cholesterol esters, like this, thinking this would be a specific substrate. And we could measure the activity, the hydrolysis of this by mass spec, because we can do mass spec on anything. It doesn't have to be fluorescent. But it turns out these substrates are hydrolyzed pretty slowly I wouldn't say slowly, just we cannot detect the activity of these guys in a dried blood spot. There's not enough lipase. The trick behind this 4-MU palmitate is it's a hand grenade. It's a super reactive ester that allows you to have very high activity and see the lipase in a dried blood spot. At the same time, it's very nonspecific, so you see other lipases. But you have to use a really active substrate to see the activity in a small dried blood spot. So anyways, we made a library of analogs of this fluorescent substrate. We made about 200 compounds. And we just said, look, if we can find one that's hydrolyzed well by the blood and is 100% inhibited by the LAL-specific inhibitor, that is a LAL-specific substrate. And we found one. And it works. So here is a bunch of, it's a single assay. We don't have to do this plus and minus inhibitor game. So children less than 5 years old, 15, 12 adults, and 6 woman patients. You can see the separation with a single assay is really good. This can be done by mass spec or fluorescence. Take your pick. So we found a specific substrate for the lipase using kind of a library approach of just getting lucky. Now, just to end, I want to talk about something that's really exciting, and Andy's going to like this. So newborn screening and proteomics. Proteomics is hard. You're trying to find the abundance of a low abundant protein in a complex mixture like blood. So the ultimate needle in the haystack problem. And so, just typically we would do this by ELISAs, sandwich ELISAs. And there's all kinds of problems with that. You can talk to Andy about thyroglobulin and autoantibodies and interferences and problems in misdiagnosing thyroid cancer with Delphia type autoantibody, ELISA antibody assays. It's hard to really prove that your immunoassay is really selective, right? The antibody could be binding to other things. On the other hand, mass spec is more selective because you're seeing it's got the right retention time on the chromatography column. It has the right parent mass. It has the right fragment masses, but it's harder to do, and it takes more time. So we got to fix that. The other thing is, well, let me just jump to the case of Wilson's disease. So I'm very excited about this project with Sihoon Han, Seattle Children's Research Institute we just started. So this is a disease caused copper buildup due to mutations of the ATP7B gene, which is a copper transporter in Golgi membrane. So we have obviously no way to do a biochemical functional assay of this metal transporter in a dried blood spot, right? That's off the table. The only thing I can think of is to measure the protein abundance. It turns out, as far as we know, most Wilson patients have mutations that render the protein unstable and the abundance is low. Is that every Wilson patient? Probably not, but most of them. So we would miss a few percent, perhaps, we don't know exactly, of the Wilson patients that produce stable, abundant protein that is non-functional, because we're going to measure the abundance of the protein. But getting 97% of the Wilson patients is better than nothing, right? All right, so why don't we do a Delphia type ELISA, because it doesn't work. You can try. We've tried. It doesn't work on low abundant proteins in dry blood spots. These antigens are denatured. It just doesn't work when the protein is in very small amounts in a very complicated mixture. So the only way is mass spec. Okay, but it's not trivial because there's many different proteins in this blood sample, and we got this big needle in the haystack problem. So what we can do is the following. We can treat the dried blood spot with trypsin to digest all the proteins into the tryptic peptides. So proteomic techniques work well on tryptic peptides, not so well on whole proteins. So we chop the protein into little peptides. Okay. And then the problem is you've got 30,000 proteins, and now you have 3 million tryptic peptides, and you're looking for 2 of them. So you got a big needle in the haystack problem. If you put it into the mass spec, you can do everything you can think of. You'll never find those peptides for low abundant proteins unless you're incredibly lucky. So we have to enrich. So we use monoclonal antibodies that bind to the 2 target peptides from the Wilson protein. And we pull those peptides down with magnetic beads. And even then, it's still a mixture of peptides because the antibody enrichment is far from perfect, and we got a gazillion peptides. So then we put it into an LC mass spec, and we look for the signature peptide from the Wilson protein that has the right retention time on chromatography. It has the right parent mass, and it has the right fragment mass. And that is, in fact, the Wilson peptide from the Wilson protein. And we do 2 such peptides as a double check, and it works. The problem is it takes about 20 minutes per baby. And so the goal of the next few years between me and Sihoon's lab is to get this down to 2 minutes per baby. How are we going to do that? To make a long story short, we have some tricks up our sleeves. We're going to use tagging, so every baby gets a different tag. So then we can do, if we do 10 babies at once in 10 minutes, and we tag them differently with mass signatures, we do 1 baby per minute, 10 at a time. All right? We just can't mix up the tags so then we get the wrong baby. But that's always possible in newborn screening. So we're going to use tagging strategy, and then put a bunch of babies together into the same 10-minute run. Okay, and that gets you down to 1 minute per baby. And that's the goal of the next 5 years. Summary, I think I'll skip that. Just to thank a lot of people involved. So, you know, the story started about 15 years ago when my wife was pregnant, second child, amniocentesis, and I asked a lot of questions like, what are you doing? And they said something like spina bifida and Down syndrome, and I saw that movie Lorenzo's Oil, and I thought, what about all these diseases? And I'm an enzymologist, so why don't we screen for everything at once and just get it over with? And then Rona told me, no, it's not that simple. And then I met Ron, and Ron said, this is a good idea, let's have a go at it. And we should work on lysosomal storage diseases because this is the next frontier of expansion of newborn screening with these new treatments. This is where the field is going. So that was, Ron was good for that. He whittled it down to what was needed in the field. And here we are 15 years later with an FDA-approved kit for doing this. 30% of the United States is live for this. The rest will come in the next 5 years. All of Taiwan, Australia, Japan is starting. Let's see what happens. A lot of people involved in this work. I want to call out a few people. Sophia Masi developed the lipase assay. She just got her MD PhD in our program here. Joyce Lau is from Taiwan. She's sitting back there. She's now in the clinical chemistry fellowship program with Andy. One of the best graduate students I've ever seen in my 35 years here. She's the one that, and she spearheaded this Taiwanese lysosomal newborn screening program, which is the most progressive newborn screening program in the world. So the only graduate student better than Joyce is Xinying, who's sitting back there. She's superstar, and she's like equal superstar, let's say. These are the two of the best graduate students I think I've ever had in 40 years I've been here. Xinying has developed the Megaplex and a bunch of other things. There's a lot of other people involved. I just want to call out these are phenomenal graduate students. I think Xinying wants to go into a fellowship clinical chemistry program also. We're talking about that today. Here's some people. I'll just end, meeting the families is kind of interesting. That I have, what these families endure with these diseases is just unbelievable. And they go around thanking us for all of the wonderful research we do. And I just wonder, I don't know, my life is good, my kids are healthy, and yet they go around thanking us for all the, and we get their thanks too. And I just want to give all my thanks back to them. And these are my heroes, right? What these guys go through is unbearable to me, and we have to do better. So newborn screening, I think, and gene therapy and things like that is the only way forward. And we can talk about how controversial this is, or we could do nothing. And I don't think we're going to do nothing. All right, so we march through this controversy and we see how we do. Okay, thank you very much.
Thanks, Mike. It was great. I don't know if you can hear me. I'll just yell.
Yeah.
So you have an FDA-approved kit, and you want to add the next couple of diseases. What do you got to do? You got to go all the way back to square one?
I guess so. But I would say yes and no. 95% of newborn screening labs are okay with LDTs. So there's a couple of states that require an FDA kit. They'll be behind until this and that. Yeah, it's kind of a problem, but not really in the newborn screening world. They're used to LDTs. As you know, if the FDA shuts down LDTs, people like you and newborn screening labs have major problems because we have hundreds and thousands of these rare disease assays for which no FDA kit is in sight because it costs $3 million to develop a kit that's going to be used 300 times a year, right? It's just not possible. So let's hope that we don't shut down LDTs. And in newborn screening, LDTs are a big thing, more than you think. So I think it'll be okay. But, if the market is big enough, PerkinElmer and other companies will spend the money for an FDA-approved kit. That's what happened for LSDs. It took a while. They didn't want to do it. I think they finally did it. They didn't want to spend the money. till the market, but the market until you spend the money. It's like a catch-22. Eventually, they spent the money. It's a big problem, but labs will do LDTs. LDTs is the way out. Let's hope for LDTs. You all know about LDTs. They're a vital thing in what we do, and it's impossible without them. So, okay.